All right, so with that, I'll ask you guys to sit back and enjoy. Hopefully, have some good technical discussions here for the next couple hours. And I'm going to go ahead and turn it over to our first presenter, Sandy Howland, with New Jersey Department of Ag. Sandy, if you're ready.
Yes, I am, Evan. Thank you very much. Thank you for organizing this, and happy to share our New Jersey Manure Link project.
Looking forward to it. Thanks, Sandy.
I do want to say, in case anyone is super excited to see this for the first time and tries to go to the website, Rutgers is having a major data server power outage today. I fully blame the fact that I was going to present on the website and give you guys a wonderful demo of it for their outage. They're currently trying to get everything online from their Wi-Fi to student emails. So won't be quite as exciting of a demo, but just wanted to give you a heads up. Typically, it's available 24/7, 365. So with that, our New Jersey Manure Link project looked at some, I guess, known barriers across the state, starting with the fact that a lot of our landscape is made up of predominantly small acreage farms. Many of them, especially our livestock farms, have limited capacity to use manure on the farm. And horse farms in particular tend to not have associated cropland and view manure as a waste. Looking at that, there's a great need for nutrient transport, moving that material from areas of excess to our crop fields, composters, urban farms, really connecting those farms with a cost-effective soil amendment. This project also addressed a major goal in our Department of Environmental Protection's Global Warming Response 80/50 report to reduce agricultural organic waste materials that are routed to landfills. One common practice at many of our horse farms is to use a dumpster service. And our final goal was to provide education and increase the adoption of composting practices on farms. To do this, we partnered with Rutgers for website development and the New Jersey Composting Council for education and outreach. We sought to develop our website to connect producers with manure and compost. We also hosted educational events in 2024 and 2025, adapt practices and develop educational material that were really focused on New Jersey specific farming, and assisting producers with implementing composting and nutrient management practices through technical assistance. So I'm going to start off with a review of what is New Jersey Manure Link website. This is our landing page. From here, you can opt to create a login or view available manure, available compost, or a resources tab. Our available manure listing defaults to a map view. This initial screenshot was from early on in 2024. You can see there's a handful of listings. By September 2025, we had listings across the state, and it really is growing every day. You can also view this information by list, filter it by county. It's very user-friendly. If you click on one of these drop pins, you'll get a quick pop-up with some basic information about the available product. If you want to know more, clicking on details, we get into the weeds. How much manure is available at that farm? Is there bedding in the manure? What types of livestock are contributing? Is there delivery? was pickup an option? Have they completed a manure test? Many of the listers have chosen to hide their contact information, which is a phone number, email, behind the login. Creating a login for the website is very simple. It's just a name and email. But as people start to use the website, that is something they need to understand. The available composting listings are a similar map format with details, and those details go into a lot of the nitty-gritty of compost, as well as what testing has been completed, how has that compost been managed, how many months or years has it been cured. Beyond our listings, there is also a resources tab, which includes information from our state animal waste management regulations, food safety and the use of manure and compost in that setting, how to compost, some information about NRCS programs, as well as listings for manure haulers. Our webinar recording is also available through the resources tab. And just a quick update, since I can't show you live, we are up to 311 users as of April 2026, with 42 manure listings and 5 compost listings. This is pretty significant growth since October. We're up about 65%. And as you can see, the breakdown of listings is dominated by horse manure, but there are a variety of livestock manure types there. And I do want to highlight that while our compost listings are low, it is, I think, truly composted material that has been temperature monitored, cured, and is ready to go. And want to highlight a success story. Charlie West, who's the owner operator of West Farm Nursery, had approached us at an in-person event and was happy to share that he had used Manure Link to source not one but 2 local manure sources, and was very happy with the tool, and felt like many were still unaware that it was a resource that was out there. So always nice to hear that your tool is working for someone, and hope that we continue to grow as a resource into the future. Moving on from the website, we're going to talk about our educational opportunities through the grant. Couldn't have done it without our partners. In addition to New Jersey Composting Council and Rutgers, our field day hosts were Copper Creek Nursery in Kingwood, Hunterdon County, and in 2025, Whitetail Farm in Lambertville, also Hunterdon County. We had assistance from John Clutie, KES Environmental, for some nutrient management pieces, as well as Michael Verbotnik from Greenway Environmental Consultants for manure spreader calibration and nutrient management. And of course, we couldn't have done it without the SIG grant itself. Our webinars, we had scheduled them 2 weeks prior to our field days in order to get that classroom type learning out of the way. And we covered a range of topics, starting with quick overview of the website, then digging into the animal waste management regulations, whether you were a livestock farm or going to be importing manure, so you were raising awareness. From there, we really dug into composting fundamentals, the methods and processes of composting, compost utilization on farms, as well as a little bit about different technologies that are available, composting economics, markets, and testing. Of course, we highlighted on-farm nutrient management, as well as resources and assistance opportunities. The webinars in 2024, we had 25 participants. That recording is available on the Manure Link website. And in 2025, with 22 participants. Then our field days, some fun action shots. This is 2024 at Copper Creek. We did hands-on stations for moisture content, temperature, and oxygen. The bottom picture are attendees doing bulk density sampling. Then in 2025, we looked a little bit more at use of compost on farms in a pasture hayland setting, looking at how you know when your compost is done, then also doing a live manure spreader calibration demonstration. Across all of our events, we had 92 participants. And we were also able to get certified to offer nutrient management credits for Pennsylvania and Maryland. The final piece of the project was adapting practices to New Jersey farms and developing technical assistance. Through a partnership with SCUMA, and a shout out to Eric and Nathaniel if they happen to be on the call, we were able to complete technical assistance guidance and basic engineering designs for manure and composting storage structures. Developed producer-ready materials for basic plans, basic site preparation, a materials list for the project, and estimated cost at the time of developing the document. And the focus of this is helping our small farms be able to move from kind of a interest in a practice to the potential for implementation. We see a lot of them maybe not understand how to see it through. So we felt like this document really offered simple, practical, lower cost options. And happy to say that we have 3 farms who have expressed interest in field testing these plans, and we'll be happy to report on that hopefully next year. So I want to talk a little bit about the SIG as a springboard. The New Jersey Manure Link project helped us identify the next gap. While we were finding many listings across the state from manure, the feedback we were getting is it's difficult for many to get that manure and get it on where they need it. So with that, we were able to develop and launch our first nutrient management assistance grant. It offered applicants funding to purchase a manure spreader, as well as establishment of a community use program. It's required for them to share with at least 5 farms. It also requires the development of a nutrient budget or nutrient management plan for those farms. And very happy to report that as of spring 2026, we have 3 grantees up and running. And we have 1 high capacity spreader with Zhang Farms, small capacity spreader with NOFA, and a medium with Fairgrown. Zhang Farm was first to get up and running in the fall and was able to report back that there was a lot of interest. These opportunities are also listed on the Manure Link website, and we hope to see each of these awards helping to continue that nutrient transport implementation. And also just want to highlight kind of continuing this work that we do have across the state animal waste management plan development and BMP implementation grants with our soil conservation districts, Upper Delaware, Del Atlantic, and excited to say coming summer 2026, Morris County. And as these grow, we feel they continue the work in this SIG and support the implementation of nutrient management, animal waste management, handling, and hopefully this all kind of this road we're on kind of continues to be successful for years to come. So with that, can I answer any questions for anyone? And
yeah, thank you, Sandy. That was excellent. And we will open it up if there's any questions. I don't see anything in the chat, but if you want to put something in the chat or raise your hand if you have any questions. I'll just say one of the things I love that you point out, Sandy, is that I've seen this historically with CIG, is that oftentimes, the initial grant gets you started, and then there's this really neat kind of spinoff that happens as it kind of grows, and it kind of what I see here too. You've really grown this out from the initial thought of just a website and some education, to some other really cool programs. So really nice job on that.
Absolutely, Evan. It was a great opportunity, and I thank NRCS for that. And yeah,
that's good.
Thank you.
Mm-hmm. Okay, so I'm not seeing any questions from this. And again, if you think of something else, we are going to have kind of some open time at the end too. Oh, I see Edwin's got a question. So I thought you were going to go off the hook, Sandy, but I guess not. You got a question. So.
Hi, Sandy. Thank you for the presentation. It was a very good presentation. My question is, when you showed that composting facility, can you talk a little bit more about that? And is that the intent of the CIG on including the composting facilities for us to try to implement that as with NRCS? And is something that was, for a specific size with idea is like could be like dynamic, if depending on the number of animal units.
Mm-hmm. The I guess document that was developed here was part of what the department was bringing to the table as our technical, and what we developed as our technical assistance piece. The hope here is that it could be a initial step that a farm takes, whether it be on their own, whether it be with a district, whether it be leading them to NRCS for manure storage. What I've seen often is that farms are quite interested in a concept, but they're not quite sure how to move forward with it. So Edwin, I guess it could be complementary, and I think we'll find out how, we'll find out more as these 3 farms give it a trial run.
Well, thank you, Sandy. Yeah, it's, that because, my idea of, CIGs are to develop things like this, right? Something that we can use and provide more options to our producers, and how we can use this also include potential financial assistance for the implementation of this. So it was very interesting to me to see that you included a component like this in the CIG. Thank you.
Yes, absolutely. And I think I skipped a piece of your question, which was, is it tied to animal units? And short answer is yes, it is modular and able to be scaled.
Excellent, thank you.
All right. Well, thanks for that question, Edwin. That was good. And yeah, all right, Sandy. Well, it looks like I don't see anything else at the moment, but you're planning on hanging around for till the end here, Sandy?
Yes, looking forward to hearing about the other 2 projects.
Cool. Good. So, like I said, if you think of anything else, you'll get another shot at Sandy at the end. Or I know Sandy flashed up her contact info too. And actually, Sandy, I don't know if you don't mind, maybe stick it in the chat too. Would that be okay if they had a question they think of later? Yeah.
I'll stick that in. I'll stick the web address. And again, apologies on this outage. It is rare. It sounds like Rutgers is having a rough day.
Yeah. And I will echo what Sandy was saying too. To me, I kind of got periodic updates, obviously from Sandy through reports and also just talking with her, but it was really neat to see the growth of that website and how quick it happened. So it's nice to see it. It definitely seems to be getting used, and was exciting to us as a staff. I know when we reviewed the CIG to get at some of these issues we're seeing, with particularly with the small horse farms and the use of manure. So that was one of the exciting things for us with Sandy's CIG application. So good. All right. So with that, if John Zander's ready to go, I think we'll move it over to John. And again, we're going to kind of shift gears, totally different directions. So it'll be neat to kind of hear. I don't want to spoil anything, but see what John's been up to with his CIG. Again, what's cool with this is it's actually a producer, which we love. We love when we get producers that are involved in programs like this. So John, if you're there, I think I saw you.
Yeah, I am, Evan.
Good, beautiful. Just want to make sure. And if you're ready, I'll turn it over to you.
Thank you. I'm gonna just pull up my PowerPoint. Okay. So I'll just go into a little bit of background about my farm, and then I'll get into a little bit of what spartina, patens, and salt hay is. We're located in Bridgeton, New Jersey, in Cumberland County, right on the Delaware Bay and Cohansey River. It's a pretty remote section of the state. The farm has been in my family for over 30 years. It consists of crop fields, woodlands, tidal salt marsh, kind of all mixed up and together there. Past crops included corn, grass hay, soybeans, cattle, and then salt hay. There's actually a history of salt hay production that goes back on the farm to the late 1600s. The present products include salt hay. We're kind of using some different methods than what were used several hundred years ago. Native salt marsh plant plugs, native trees and shrubs, and then we just started producing salt hay-based biologs for erosion control and restoration projects. And here's just a few photos of some activities around the farm. The upper left there, that's a couple of my interns from the project, gathering material for plugs. And then just some salt hay harvest going on in not the cultivated plots, but the naturally occurring salt hay plots. And then just a little bit about salt hay for those that don't know. It's they've used 3 different types of salt marsh grasses in the past, but the most desirable of which was spartina patens, known as salt meadow cordgrass. Grows in the high marsh areas that are not flooded as frequently as the lower marsh areas where the smooth cordgrass occurs. Grows in thick mats in lengths of 12 to 36 inches, and it's kind of a monoculture of just that. You'll see some saltgrass mixed in occasionally also. It's naturally occurring, completely sustainable. It needs no intervention from us to grow every year. Although when we planted it for the SIG, we kind of came up with some different things we can do to manipulate the growth a little bit. It can be harvested twice a year if you time it properly. We don't typically do that. We try to avoid the critical nesting periods during the summer. We typically harvest most of it during the fall and winter seasons. It's very long lasting and rot resistant. It's got a multitude of uses for anything from garden mulch to packing. In the past, it was used for one of the weirdest uses was casket bedding, I guess. It was used for packing glassware, making rope, all types of different things. The photo just shows how it was harvested way back when, and the methods that we use aren't all that dissimilar from that image there, unfortunately. Just a few more photos from the salt hay harvest on the naturally occurring plots. So we use boats to haul it in often. The photo on the right shows the length of the grass compared to a foot. We use tractors with turf tires and light machinery, so we're not kind of damaging anything out there and leaving imprints on the marsh. It's a pretty harsh environment for equipment. So then we get to saltwater intrusion. So we had areas of the farm that were in ag production for years, and they started to get frequently flooded, not just around storms and like storm surges and hurricanes and things like that, but just on a normal high tide, we'd get flooding in a field. The photo there actually shows what was a cornfield and hayfield adjacent to a marsh that just flooded pretty frequently, several times a month. And when we experienced these floods, it would creep in and kill off all the crops. So we really didn't have, there was a bunch of areas in the farm that were now rendered useless for production because of the saltwater intrusion. So we started kind of thinking about what can we grow there instead of trying to fight these conditions or roll the dice and plant and think maybe it won't get flooded. So we looked to the production of salt hay on the farm. We thought, well, if we can plant some salt hay in these areas and get it to take, it'll be a lot easier to access with machinery. It'll restore these unproductive areas. So here's another photo of one of these fields. This was actually a cornfield that was in production probably until roughly 2007, 2008. It's adjacent to a marsh, and this is just a normal like daytime high tide. I believe it was a full moon tide cycle. Just kind of shows you really can't do too much there as far as traditional ag crops. So we were trying to think about what we could do with these areas that just were void of production, and we, as I said, we went back to salt hay. We did some experimental planting prior to the SIG, just very crude, taking pieces of salt hay that grew kind of along the roads of the farm where maybe they fell off the hay wagon or whatever and started to just produce on their own, and transplanted them to some of these areas. And we saw some success, and that led us to apply for the SIG. And once we got the SIG, we started to develop site selection and prep. It was pretty easy. I mean, you could just go on. I mean, just from experience on the farm, you can kind of tell which areas had saltwater intrusion just based upon the soil and what was the lack of what was growing there. And then some of the goals throughout the project were site selection and prep, donor plug harvest method. So trying to figure out where we could get these donor plugs. One interesting thing that was pretty serendipitous during the first week of the project, we were kind of looking around for some donor plugs, and we happened upon a county work crew that was doing some road work, and they dug up a whole bunch of patens and just kind of dumped it that was growing next to the road. They just dumped it in a heap. And I think we were able to get it onto a trailer and bring it back to the farm. And I think we made like over 30,000 plugs or close to 30,000 plugs out of that material that would have just gone to waste. So we were pretty fortunate to come upon that. We experimented with different planting techniques. We did some planting with machinery that we kind of adapted to the purpose. We did some manual planting because oftentimes you couldn't get machinery into some of these areas where we had planned to put some plugs in. We monitored EC. We quickly found that changed almost daily, just depending upon the flooding and the conditions present there. And then harvest methods and timing that to try to figure out how to maximize efficiency and yield. And then also the application of our methods that we're developing on the farm to other coastal farmers. There's quite a few farmers in my area that once they heard about the project, they were pretty excited about it. And then just some photos of our planting. So this just kind of shows using the tractor. This is the second photo there is a little bit after, I guess it was about a month after initial planting. The upper right shows that cornfield that was in a couple slides back there. That was prep before we put plugs in there. And then in addition to plugs, we put in some mats that are shown in the bottom there. So just to try to see what would what would take better, whether it was plugs or larger mats. And then we also experimented a bit with spacing of plugs. And then some of the challenges that we faced were working around tide cycles, and not just tide cycles. So if you're familiar with the Delaware Bay area, the wind has quite a bit of an effect on the tide. So we had days where you would expect to have like a low tide and it would be pretty dry, and all of a sudden the wind blows out of the northeast and we're flooded out. The equipment, we had constant equipment failure, getting equipment stuck. Just the environment with the salt water is really rough on things. Biting insects, we had every possible insect out there just tearing us up throughout the process. We used head nets when we could. That didn't really work for the gnats too well. It was really tough to get labor and interns on the project just because of the harsh environment and things like the bugs. It was often hot. It was just a constant battle throughout the project, but it was very rewarding at the same time. And then there was a constant fight with Phragmite. It kind of likes the same conditions that we were planting in, and it was constantly creeping into our plots. So we were kind of trying to come up with ways that didn't use herbicide to control the frag. And some of the things we did were mowing high above like as soon as the frag kind of crept up above the height of the patens, we would kind of mow it back. Using a variety of mowers, everything from a flail mower set pretty high to a traditional rotary mower. We used a sickle bar mower on a 2-wheel tractor, and all those methods they're pretty efficient. It's just you have to continue doing it and keep up with it. This year we're actually seeing in the plots a lot less Phragmite, so it seems like the patens has outcompeted the frag. Just some more photos here of some of the challenges. So there's the tractor stuck there. Coastal flooding that we experience all the time. You can see the guys planting there with the head nets on for the bugs, and another stuck tractor. So some of the benefits that we were looking for in the project were to increase the production of live plant plugs to meet the growing demand for plugs in wetland and coastal restoration projects, to restore productivity to other coastal farms that were experiencing the same challenges that we were with saltwater intrusion, create enhanced habitat for wildlife species. It's a lot of these plots, even though they're cultivated, they just look like they look very similar to the naturally occurring plots. So we've seen all kinds of wildlife taking advantage of that. To slow the spread of Phragmite within these buffer zones, we've definitely seen that with the mowing. One thing I didn't mention, another reason why we were kind of against using herbicides, was a lot of the salt hay mulch gets sold to organic farmers, so we try to avoid using that stuff anywhere even near the hay. And then also to hopefully slow the spread of saltwater intrusion. That's kind of the jury's out on that one. It's really tough to measure that, but I definitely can see it looks like everything that we've planted is really, this year especially, is really taken off. So hopefully we'll see that effect. Some more photos of some of we actually were able to harvest some of the hay from the experimental plots last year. So and some of the plugs as well for use in restoration projects. This was the upper left is a project in Maryland. We took some to the Edwin Forsyth Refuge. That's not our hay in the pictures, but we delivered some there to that project site. There's a group in the upper right from one of the many farm tours that we've done, and then just another photo of our planting. And then some of the key results and facts from the project. We planted over 78,000 live plugs, and they were everywhere from like 2-inch standard plugs to mats to 4-inch plugs, 6-inch plugs, on just over 6 acres on 12 different sites throughout the farm. We developed novel plug prep methods using different tools, everything from like crude methods with knives and things like that to manual methods and machinery. We identified key planting conditions, or like I said, they're not all that difficult to identify. Produced a technical paper, kind of still finishing that up now, along with a guide on the practices that we used, and kind of got a typo there, but we gave 17 on-farm tours throughout the project period, and we've continued to do them presently. I gave 9 off-site presentations on the project, and then we had the project highlighted in 8 media publications. We're presently developing the use of our Swizz ag techniques for other species that thrive in these types of conditions. And then actually just recently I had a group of grad students from the Netherlands contact me looking to learn some of the techniques that we used. And of course, in addition to that, we're working with some of the area local farmers that are experiencing the same saltwater intrusion to try to get them to catch on using some of these techniques. It's been a little bit tough, but there's a lot of skepticism, but I think I think we're starting to make some headway there. And that's basically it.
Awesome, John. I got a couple questions coming in here, so we'll see on the chat here. And maybe before I just jump into questions, I kind of just like to give some insight into, this is another, again, state CIG, and so on these state CIGs, we kind of develop technical teams, to review the applications that we get. And this one was just really cool. It hit a lot of the marks that we're looking for. It was working directly with the producer, which was neat. Saltwater intrusion, something I hadn't spent a lot of time in my career in John's particular area of the state, and going down to see him and visit him and see the impacts that they're facing down there is pretty startling. If you've not found yourself, like John said, he's kind of in a more remote area of the state, and you can really see the impact. A lot of nice overlap with our PMC's efforts. I know Chris Miller, who's on my staff, heads up our Cape May PMC. They were doing a lot of work on saltwater intrusion, so he was really excited about this too, and the applications for a native plant to be used, like John mentioned, in restoration activities or expand markets. I think producers in Delaware and Maryland, a lot of them facing similar challenges here. So it just hit a lot of buttons. It was a really good project. It's great working with John, and it was really exciting to see some of his results there. All right, so going over our first question here is from Don Donnelly. He's our state forester here in New Jersey. John, can you talk more about the EC monitoring you mentioned? Any findings and implications of importance from that?
So we basically just use like EC surface soil monitoring, and like I mentioned, it was really all over the place, just depending on the tide. There really wasn't a lot of consistency with it. We kind of more shifted toward just visual identification of planting sites, just based upon going out and seeing where this tide was kind of flowing in. Not so much. I mean, my interns monitored all 12 sites daily for quite a while, and there was really not a lot of consistency in the data. It was just more qualitative selection of, okay, there's no upland plants growing here. It's more, there was a lot of frag creeping in. You could use that. But like I said, you could just kind of watch the tide, and around some of these higher tides, just sort of figure out where it was coming in. So that was more what we used. I think there's definitely a lot of opportunities for other coastal farms, but I know salt hay is kind of like a forgotten thing that comes back, and people are like, what the heck is that? And then some people are like, well, I remember that for, when I was a kid, we used it for bedding for our cattle, or we use it in our garden. So it's definitely something that we're trying to bring back, and there's been a bunch of interest in it. So I think there's for sure some potential for other coastal producers.
Yeah, I think you even said, right, John, one time I was talking to you, you had a little market going with even like duck blinds, right? Or duck boats. People were buying salt hay off it. Just interesting markets out there.
Yeah, we have like a little stand out by our main gate where we just leave bags of salt hay, and we just sort of started doing it, I guess, 2 years ago. And then actually, it's funny. Last year we cut some of the hay from our sig plots and put it out there with some of the naturally occurring salt hay side by side, and that went first. It was more robust, it was longer, it was thicker, and that's actually what has kind of gotten some of the local interest in producing salt hay, similar to what we did. Just seeing the increase in traffic volume on the road coming down from the duck hunters in the fall to pick it up. I mean, we've had days where we put out over 100 of those bags in some of the photos, and they're on a Friday afternoon and they're gone by Sunday, just from duck hunters using it for their boats. So it's kind of a niche market, but it's helped to raise awareness, because some of the other neighboring farms are kind of like, what the heck are all these cars coming down here? We used to see maybe like 10 in a day, and now we're seeing like 100 in a day. So it was kind of cool to use that to sort of raise some awareness for the project.
Yeah, very cool. We have a second question here from Virginia Lamb. John, can you talk more about the amazing wildlife presence that has evolved on your farm as a result of your practices, eagle pairs, etc.?
Yeah, so we actually have, I had some more kind of more fun facts in the beginning of my presentation that I opted to take out just for time. But we have 8 active bald eagle nests on the farm right now. And they're just really thriving, and there's I mean, it's just teeming with wildlife. There's everything you can imagine that South Jersey has on the farm. But the eagles are definitely kind of the stars of the show. Like we would bring people out. Like I had a tour out, I guess it was about a week ago with the local watershed association, and I had the whole SIG display set up, and I had plant plugs on the table, and they were right within sight of a nesting pair of bald eagles, and everybody, it was like I was fighting for attention with the group from the Eagles. So everybody's kind of turning around to watch them, and not so much listening to me. So it's kind of cool. But yeah, we have a ton of wildlife, and a lot of our practices are kind of tailored around the wildlife. Like I mentioned, the mowing of the hay, we try to avoid the critical nesting periods, and really everything that we do as far as ag practices kind of meshes with wildlife management as well. So that's really at the forefront of our activities.
It's good. Thank you. So that's it from the chat. We'll give it a couple more seconds here if anyone has any other questions. Edwin.
Hi, John. Thank you for the presentation. Also, it was very interesting. In addition of electric conductivity, do you have a chance to measure other properties like organic matter or pH or anything else?
We measured pH quite a bit as well, and that was kind of all over the place too. After about 2 weeks, we just sort of relied more upon just qualitative identification. And maybe that was just kind of from my familiarity with the farm and over the years kind of seeing what didn't grow there. But we did have a couple of plots where the pH was off quite a bit, and that it was just also at the one season we were kind of dealing with that drought, and that kind of dried some areas out quite a bit. But yeah, it was more just very quickly we kind of adapted to just sort of almost like you could just look at it and tell this is a good place for it to grow. And even when we've kind of continued with some other species that we put in, we don't really do a ton of technical analysis anymore. It's just more kind of going by feel, which I realize is a little bit crude, but it's kind of been pretty effective for us.
Thank you.
All right. So must see anything else on the chat? Any other hands? John, are you sticking around to the end here?
Yeah, I'm going to stick around. Sure, I'll be here.
Okay. And same thing, John. It's up to you if you're comfortable putting your contact information in the chat if people want to reach out to you or whatever.
Yeah, absolutely.
Oh, yep, that would be great. And again, nice job. Thank you for that.
Yeah, thank you. And thank you for the experience. It's been outstanding. I really appreciate it.
Yeah, it's been excellent. It's good. Okay, so last but definitely not least here, we have Erin McHale. She's a PhD candidate with Rutgers University, and I believe Dr. Maslo is on too, kind of in the background too. So, and they're going to talk about Rutgers CAG that just recently got completed.
Hello, hi everyone. Just going to share my screen here, and I'll also save bandwidth and maybe turn my camera off as well in a moment. Forgive me, we usually do this on Zoom for Rutgers, so I'm learning how to do.
Yeah, well, that happens to me every time we're on Zoom because I'm used to Teams, so I totally feel
Right?
you.
All right. So, can you just let me know if you can see this?
Yep, it looks good.
Fantastic. Okay. So first and foremost, thank you for having us here. We were really excited to be able to present the project we've been working on for a few years now. Just a side note, forgive me if I clear my throat sometimes. I'm getting over a bad bout of bronchitis. It's lingering a little bit, so hopefully we don't have any issues there. But my name is Erin McHale. I'm a PhD candidate at Rutgers University, New Brunswick. I work under Dr. Brooke Maslo, who is here present. And this project is thanks, of course, to the state grant that we received. And without it, this wouldn't be possible. So this project, just to set the stage here, this project is one of my PhD chapters for my dissertation. And my overarching theme of my dissertation is biodiversity. So when we had this opportunity to get this funding, it really took off a lot of our ideas, and we were really excited to move forward with it. So I'm excited to present that work to you. So the overarching theme here, again, is biodiversity. And I know most of you are familiar with the term biodiversity. Why is it important, though? Why do we need biodiversity in both agriculture and just normal environments across the world? So this is a very broad multi-sectional project. I'm going to try to streamline it as best I can. So if I gloss over any part of this project or any of these slides, definitely ask me after the talk, or I can, provide my email as well. It's at the end of this presentation, and I'll be happy to clarify anything that I'm saying now. So biodiversity, there's a lot of ecosystem services that we enjoy as humans from animals in general, especially in agriculture. For example, pollination. Having multiple animals on your agricultural land can provide pollination for your crops, for the environment in general. Having different microbes and different plants on your farm can create nutrient-rich soil, which benefits you in the following season. And then the one that we're going to kind of focus on today is pest regulation. Having animals in general in the area that you're on, especially in agriculture, that can ingest pest species is beneficial. So we're familiar with biodiversity, giving us that service of, providing these benefits to us as humans. However, biodiversity in general across the planet is important for a lot of other broad reasons as well. One is ecosystem health. Having different plant species, different animal species can promote a healthy ecosystem. Ecosystem resilience, for example, having different tree species, especially in mangroves, on edges of swamps, can create resilience against flooding and storms. Having just one species usually it will create higher bouts of flooding. And ecosystem stability, this is just one example, having herbivorous species in a grassland eating different types of grass can benefit the overall ecosystem by not allowing one plant species to take over the other. So I say all that to say, in this project, we were interested in looking at one particular mechanism of biodiversity, where it links biodiversity to ecosystem function. And when you talk about biodiversity in a landscape, there's lots of different ways that species can interact in that landscape. But two ways that are pretty cool and pretty interesting to look at that we chose to do for this project are functional complementarity and functional redundancy. These two situations are always ebbing and flowing, and they're never static. They're always different throughout seasons, throughout time. But these are just the two completely separate explanations. So functional complementarity is if you had, for example, two pollinators, different species on your farm, for example, and they would have 4 plants. Species 1 would pollinate the first 2 plants, species 2 would pollinate the second 2 plants. If one of those species was to die off for whatever reason, climate change, habitat loss, if you lost one of those species, the other 2 plants would no longer be pollinated, and either the farmer would have to step in and do hand pollination, or that species would no longer exist. And I'm using this kind of interchangeably between forest and agricultural landscape. In situation number 2, functional redundancy, that is when you have 2 species in a landscape performing the same functions all around. And it might seem to, the non-expert, well, if you lose species 2, for example, you're going to have, species 1 can just take over and perform the rest of the functions. And it might seem that way, but if you lost any species in that situation, you're going to now put a strain on species 1, and the function being performed, whether it's pest suppression or pollination, will no longer be as successful as it was when you had 2 species. So this overarching theme of biodiversity-based ecosystem function, which is essentially looking at biodiversity and how different animals, different living organisms are interacting in an ecosystem, that type of research has mainly been done for the most part in smaller systems where you can kind of manipulate experiments in laboratory settings, plant systems outside, invertebrate systems, which are usually looking at small macroinvertebrates like worms in soil systems or insects in pollination systems. So there has been a lot of research done in these systems, but there hasn't been a lot looking at vertebrate systems and how they interact with their ecosystem. Now, you might be thinking, well, I know a lot of papers that look at, how bats interact or how foxes interact, etc. But those papers aren't looking directly at how the species are interacting with themselves and performing functions in the environment. There are a few, but it is a pretty lacking area of study. So why is that? Why are vertebrates not as well studied when it comes to biodiversity as opposed to microbes or plants? Well, these are just a few small examples here. Examples of us in the field here. This is my colleague. We're out in the field. We had tagged a few bats, looking at them to see how their movement is working. And that was a long, treacherous, I'm sure many of you have tracked animals before. It can be hard work, treacherous work. If it's a huge project, it might require many hands on deck that requires, funds of some sort. You have situations where you have long range tracking, where it's GPS tracking, collar tracking, which again is expensive. So those types of projects are less known. When you do have them, they're usually looking at exclosures or enclosures where you would cover a crop, say, for instance, to see how pollination works with birds. But you can't actually look at the bird. You can just cover your static crop and see how the pollination would change if that crop was covered. So projects like that are definitely in the literature. But we thought it would be a good opportunity to kind of look at two of our own species that we have here in New Jersey and how they interact on in agricultural systems. So the project that we're doing here is taking the fecal material of two aerial insectivores, the barn swallows that we have here, and the big brown bat. The first species that we looked at, I'm sure many of you are birders and familiar with the beautiful barn swallow, Hirundo rustica, very common aerial insectivore across New Jersey and the world. It is common in agricultural settings. It is in global decline. I'm happy to go into why at a different time. They exhibit high nest fidelity, so it's easy to go back and take samples from them each week because they go back to the same nest every year. And once they're there, they go back every week, or I can go back every week and know that they're going to be there pooping. The second organism we did for this project, we took the feces of the big brown bat. They also exhibit high nest fidelity, high site fidelity. I am more a bat biologist in training. This is my first project working with birds, and I was really excited about it. I learned so much about them, and it was really cool to see how they interact in foraging with bats. So bats, again, same situation. They come back to the same roost every year. So we know they're going to be there. We can rely on them to have their droppings, and I can go and collect it every week, right? Knowing they're going to be there for the season that they're active. And they're also well known to feed on agricultural pests. So the whole point of this project was to, again, go to farms every week and collect the fecal material of bats and barn swallows and see what they're eating. But first we had to actually find the farms. So I've always told people who I've done talks for about this. This is my favorite part of my whole PhD thus far. I was able to go out in the field and I literally went door to door to farmers. I think I met over 150 people, farmers. I went to like 150 farms. And every single farmer that I knocked on their door and said, hi, I'm a PhD candidate. This is the project we would like to work on. Even if they had no idea if they had bats or barn swallows, every single one was like, come on back, let's go look. And, I can help you see if we have that available. So super amazing people. It really opened my eyes even further to how great farmers are and how much we need them. And going to all those farms narrowed me down to just 6 sites that had exactly what we needed. And those things were a certain type of crop on the farm. We needed to have them have bats and barn swallows and a known amount, like a known capacity. And these 6 farmers had barn swallows that came back every year, and they had already put up bat boxes or had them in their farmhouse. So a reliable source of feces. So what we did to start this project, after we picked our farms, we went in, we found where the bats or barn swallows were going to be, nesting, and essentially where their poop is going to be found. And we put down platforms, which we went every week, starting in late April to early October. We went in, we collected the fecal material once a week. And then once we did that, we cleared away whatever method if it was a platform or on the concrete or in a bucket. We sterilized the area, took the feces away, and then had a new stage for the next following week where we collected it again. So we did this every week at all 6 sites through all these months in 2023. At the end of the project, we ended up with approximately 210 samples. Again, that's 2 samples from each of our 6 sites across a study period. And when you have those samples, you can't tell what's in there until you do a whole bunch of laboratory methods. And I'll just go through this briefly. I'm happy to talk about these in length if you would like to talk about it after. But essentially, you take the sample you have, you analyze it through PCR. You essentially multiply the amount of DNA in your sample, so it's easy to analyze. We send it off for sequencing, where they basically take all the DNA and make it nice and prepared for us, and then they send it back to us. We did the sequencing at Princeton University, just to clarify that. Once Princeton sends us back that nice, pretty organized DNA array, we take it and I run it through a bioinformatics pipeline. Bioinformatics is basically taking the DNA that is basically a milkshake, and pulling it apart and organizing it and making it nice and cut and clean and preparing it to take our DNA and put it against an already existing library. So if you're not familiar, there's lots of libraries that have already, people have gone out, taken DNA from individual insects and put it into this library. So their DNA is available. And then we take our mystery DNA, we feed it into this library, and it tells us, okay, your DNA, you have a wasp, a spider, spotted lanternfly, etc. in your sample 1. So now we have a nice organized array of all of our samples and what is inside each of those samples. It's essentially a menu of, this is what barn swallow ate from the week of May 1st to May 7th. So once we have that nice sheet all done, we have all of our DNA finished, we can see what's inside each of our samples. We're able to analyze, okay, what's happening here? What are they eating? Are they eating the same things, different things? It's going to get a little science heavy here, but I'm going to try to go through it a little quickly. So if I say anything that's confusing or anything, I'm happy to clarify after. So the results that we saw first, which was pretty interesting, taxonomically from order to species, these are Venn diagrams. On the left, just take order, for instance. On the left, you have bats, ate 7 orders. Barn swallows, they had 1 of their own individual orders, which was springtails. And then together, they shared 10 different orders. And that's pretty common because orders at the taxonomic level, an order is like moths and butterflies, right? It's a huge order with lots of different families and species inside of it. So we weren't surprised to see that. But as you go down to species, for instance, you see that bats had 144 of their own species and barn swallows had 109 of their own species. And together they shared 41. And this is just one project across the span of, a few months. So if this was to continue to different summers, I assume we would have, a higher number of those. I know this slide's a little bit busy, but we really like this figure. We think it's pretty cool. And it shows you the bottom half here, the black and white portion, is showing you what big brown bats and barn swallows are eating. For example, on the top, you see moth and butterflies. That's an order, right? That's a gigantic order with lots of stuff inside of it. If you look up at the green and blue phylogenetic trees, essentially, this is a diet map, just showing you that although they both overlap in moth and butterflies, when you branch out and look at individual families and species, that is where it kind of gets finer tuned and separation occurs. So they look like they're eating the same thing at the big level of moth and butterfly, but when you break it down into individual species, that is where things become finer and they actually have a separation of their diet. One of the biggest points for me, especially when tying it into farmers and telling these farmers, hey, if you have bats and barn swallows on your farm, you're actually having this ecosystem service delivered right to your door, right? So again, this is just one season. And even just in that season, we found many minor pest, major pest, and disease vectors in the diets of both barn swallows and bats. For instance, on the left in red, we have the bats that we collected feces from ate the brown marmorated stink bug, the corn earworm, the spotted lanternfly, and the tarnished plant bug, and the citrus flatted plant hopper. And the barn swallows, they shared the tarnished plant bug with bats, which is pretty interesting, but they also individually ate the cherry fruitworm moth. So even if I just showed, I have shown some of the farmers just this information that the farmers that I work with, and they're amazed. Like that's incredible. Like these guys are really helping us out, even in a small way. So I know this is a lot of words, but I just wanted to kind of really show that, hey, these guys are really eating a lot of agricultural pest that can cause a lot of damage in our farms across New Jersey. Really quickly, so we did a non-metric multidimensional scaling analysis on our diets as well. And this is a broad scaling device where it shows you the degree to how similar their diets are. The brown on the left is big brown bat, on the right is barn swallow. And you can see some separation here. So kind of the same thing. You see separation. They are eating different things, of course, but they also share things in the middle. So they are overlapping a little bit on their diets. And I've given this talk a few times to the public, and I always get asked, from a non-scientist, it would seem kind of obvious that they're eating different things. One flies at night, one flies during the day. Like obviously they're eating different things. And that's very true. However, there are insects that are active during the evening and during the day. And that does coincide with some of the overlap. And the big takeaway here is that crepuscular insects, which fly during dusk and dawn times, overlap with bats going out to feed and barn swallows going home to feed or going home to sleep after feeding. So even though they are separate species, to avoid competition, bats and barn swallows do overlap a little bit when it comes to dawn and dusk. So I have a lot of other feelings about this, and I can go into more detail, but I don't want to spend too much time here. The last result I'm going to show you, I promise, I know it's sciencey, and one of the most exciting ones for us. When we started this project, just thinking it from a broad standpoint, bats and barn swallows in the start of the season, when the insects haven't emerged yet, or they've emerged a little bit, it's still cool, it's not too humid, it's not too hot, there's less to eat. I had assumed that, I would assume if there's less to eat, me and barn swallows have to eat the same thing if I'm a bat, because there's less to eat, right? And then I would assume as the summer months went on and it became July and it was hot, we probably can eat different things because we don't have to share anymore because there's more to eat. That is what my assumption was going into this project. It was actually completely flipped. And it's very interesting because what happens is when there's less to eat, and you can see here September and May, April and October aren't included because the sample size was so low, just to clarify that. But May and September at the family level, this means insects at the family level, their diets were actually less similar in those times, meaning they probably didn't want to compete for resources, so they separated their diets. That's one hypothesis. And then as time went on and it became hotter and there's more things to eat, they converged their diets and they became more similar. And then when fall started to come about, they separated their diets again. So bats and barn swallows have an interesting relationship. Again, that figure I showed you in the beginning, functional complementarity and redundancy, it ebbs and flows, it changes throughout the season, as well as in the diets themselves. So I know that was a lot, but overall conclusions here, we already know that big brown bats, and some studies have shown that barn swallows too, are generalist insectivores, meaning they generally eat whatever they can forage. Functional redundancy and complementarity, so how they function in the environment changes throughout the season and changes at different level of the taxonomic groups that they're eating. Dietary overlap also changes and shifts throughout the season, likely tracking whatever the insect abundance is at that time. And a big important one for us, especially, the farmers across New Jersey, the world, having these animals on your farm can contribute to pest suppression, right? I'm not saying it changes the population, but definitely a key player here to lower those agricultural pests. They are eating them. So together, this is saying, hey, if you have these on your farm, it is a benefit to you. Lastly, I didn't mention this in the beginning, but I bring this into my talks a lot as to, of course, you all understand why we should care, but the general public, why should we really care about this type of thing, when it comes to agriculture? So our global food demand is expected to increase a lot by 2050. And as that changes, we're going to have to use these agricultural landscapes in, a certain manner. Right now, it's a lot of agricultural intensification where it might be bad for the land. It might not have the same crop output you would hope for. So my hope as my career advances is to maybe bring this type of ecosystem services into this agricultural land that have an ecological intensification. So having nature and agricultural landscapes come together and hopefully mitigate some of that offset of agricultural expansion. Because we need food, we just have to make sure we do it in the correct way, of course. So wrapping it up, besides our research, we did a few really fun things outside of the lab with our grant. We made 250 barn swallow platforms, which barn swallows can nest on, and I can explain that further after this if you have questions about that. So we built a bunch of them. It's just two pieces of wood together with screwed together in a certain way. We brought them to an elementary school in Colts Neck or in Lincroft, and we let the kids design their own with paint on the bottom so the barn swallows don't get scared. And then they put them up around the school, and it was really fun because they were hoping that barn swallows would nest on their platform. In addition to that, we did some talks for them. We presented on barn swallows and how important they are. The kids were really excited, and it was just a really great time for us to do that with them. They had a really great time. Besides that event, I've done a lot of talks when it comes to bats and barn swallows when it comes to this project. So the funding also has given us ways to do outreach. I recently presented at the Wildlife Conservation Society conference on this work. We created a fact sheet, which you can find on the Rutgers website, how to support barn swallow colonies on your property. Even if you just want to have one barn swallow platform, it's interesting to read that. The most recent talk I gave to the public, several farmers were there. It was really great. It was up in Parsippany. I also gave out a lot of our barn swallow platforms to the farmers that I'm working with, and a few of the other farmers that I had talked about before that don't have barn swallows on their property but are hoping to get them. And lastly, we have 2 YouTube pages. One, I'm not sure if it's working right now. It was a live feed for barn swallows. It was a live feed on the actual nest, which was really cool. And then we have the RU Wildlife Wise. There's a lot of different videos in that series, but this one was really cool. It is a barn swallow ecology and conservation video. So really, really cool stuff there. I'm sorry I talk so much. It's so hard to take all of my work and jam it into a smaller talk, which is great. I love doing it, but I tend to talk a lot. So thank you sincerely to the USDA NRCS. We couldn't have done this project without our grant, so we are completely grateful to you. My email is at the bottom here. I can also add it into the chat. So thank you for listening.
Thank you, Erin. That was excellent. And just like I did the others, I'll just speak a little bit to, again, this was a state-funded COG, so we had kind of our technical team review this. I know one of the things we thought was interesting was, there's obviously been a lot of attention to bats, and I think we all, most of us know that bats and barn swallows provide some really good ecosystem services, but I don't think we were paying attention as much to the barn swallow aspect of that, which was kind of neat. And NRCS has and I'm going to lead this into a question for you too, Erin. So NRCS has a practice standard. We can work with farmers we have a practice called wildlife structure, pay for brush piles, pay for bat boxes, it's pretty broad, right? We could also pay for potentially barn swallow nesting platforms, so we have a lot of our field staff on the call. They're out working with a lot of farmers across the state. I mean, do you have some advice or some guidance or some suggestions in terms of, how we should go about approaching farmers for these, or what's your advice for kind of what we could do with that practice? Do you see a need for that on the landscape for promoting barn swallows and bats? And
yeah, that's a great question. I love that question because even though I'm a scientist, I love connecting what we're doing to real life, real world situations, especially in agriculture. And one thing I've told a few people that have come to my talks and even my farmers is even just putting up a barn swallow platform. One of the reasons they're in decline is because a lot of farmers, understandably, are changing from wooden barns, the old wooden barns, to aluminum barns, which is good for them because it's, you're not paying as much money to get it restored or, fire hazards. So while you're doing that, barn swallows aren't able to put their nest on these aluminum barns. So something you could do to keep that service they're providing is to take one of the barn swallow platforms, either remade or you can find it online how to make a different kind. Just nailing them up in, an awning inside your barn or somewhere where we can come out and assess where's a good place for you. I'm happy to do that as well. Putting those up, putting bat boxes up. I don't know if you saw my presentation. One of the farms that we worked with had 4 bat boxes up right next to each other over their barn, and they were all occupied. And they're on an orchard. So they know and they're happy to have bats there. So putting a bat house up is helpful. Planting native plants, if you can, in small areas to promote even more like native insects will bring, bats into your property, things like that. So I think it's really important to do those things.
No, that's good. And I think when I was looking at it too, right, there's nesting cups and like nesting platforms. Is one better than the other, or
So I don't have personal experience with nesting cups. And personally, this is completely my own opinion. I think they're amazing, and I have heard many success stories. However, there is one barn that I worked in that they love their barn swallows, and they had like 50 nesting platforms, just the straight up straight side type of wood, every single one of them was occupied, and it was in a really small space. So personally, I think just from my experience that just the platform would work better, and they're easier to make if you're trying to make them with your own materials.
Yeah, it looked pretty simplistic. That's good. Good. Well, thank you for that and answer my questions. We do have a couple here in the chat. So we have Jess here, one of our soil cons up in our Mount Olive office. Are the DNA data broken down on quantity within the samples, like a percentage, or is it just a presence metric? While bat or barn, while bat or barn swallow may be eating the pest species, it is good to know if they eat a lot or only occasionally. Perhaps this is the next step for your study, but worth asking.
Fantastic question. I have gotten that before in science talks, and it's always interesting because currently there is no real way to test, okay, I have this guano sample, right? Just say it's one bat poop. And I find, one of our papers was finding the spotted lanternfly DNA in that fecal material, say one bat poop, we analyze it and we see, okay, they ate spotted lanternfly. You can do a read count, which means there's more of that DNA inside that fecal material. However, it's right now just a presence versus absence, because if we have presence, we don't know if it ate just, an antenna. We don't know if it ate the whole bug. We don't know if it ate 5 bugs. So right now, at this stage of technology, it's hard to assess, how many of the insects they ate. And that is something we'd really love to work on if it was available. Of course, thank you.
Yeah, I just want to add to that. So that one way that we sort of quantify whether consumption is occasional or a common, a common prey item is the frequency of occurrence within the sample over the sampling season. So, occasionally there's a lot of species that maybe you only find it on one day or at one farm on 1 week. And then there are other things that, you can plot it out over the course of the season that, okay, this particular insect species, we have a higher frequency of occurrence across the season. And so then we can get a better idea of, oh, this seems more of a common prey item as opposed to something that's much more rare. It is a bit of a snapshot. There are some methods where people are looking at, as Erin said, the number of reads as a measure or a proxy in some ways for biomass, but it's not something that we would feel totally confident in reporting and saying, hey, they're eating this amount or this number, equating the biomass to some number. So just want to throw that in.
Yeah.
Yeah, so I have another question, but I'm going to jump to Jess's, who just said, yeah, that's what I recalled. You can't quantify readily. Thank you. Yeah, using like time sequence to try and get at a percentage. So I was just throw feedback there.
Yeah, exactly. Yeah.
All right. And then Thomas Blake is another planner out of our Mount Olive office, asked, do you see any way to expand this to forest pests?
That's a great question. Our last paper we published in Forest, looking at forest pests using the same method we used here. But we were, no, sorry, not the same method. We were looking at qPCR, which is essentially you take a guano and you look for one insect. And in that case, it was the spotted lanternfly, which is a forest pest as well. So you could definitely expand it to forest pest in terms of using metabarcoding at looking at bats that, are prevalent in forest, which are bats as well, our bats, big brown bats. And you would just look at the metabarcoding and see if any of those pests are prevalent in forest. So definitely a method you could use in forest pest. You could use qPCR, looking directly at whatever insect you're looking for. You would just have to create the assay if it's not readily available. And you could look directly for that forest pest in feces. And you could also look for those species in the fecal material using metabarcoding. So definitely possible. And I'd be really interested in doing that as well, in addition to what we already have done.
Yeah, Evan, I just wanted to throw something else out too. So back in the day, our inaugural SIG grant was with brown marmorated stink bugs and looking at something similar in bat consumption. And the idea there was, can you, instead of the conventional blacklight trapping methods, which is what was available at the time, could you use these guano piles as a reliable survey area in which to detect invasive insect pests or emerging insect pests? And so So basically the results of that study were that, yes, at least as far as blacklight traps were concerned, we detected the stink bug in the guano about a month prior than the blacklight traps because we had simultaneous sampling. So as part of this project, we were also able to leverage some of the funding here and adding some of my own lab discretionary funding. But we're doing an additional project to look at whether or not basically can we look at the bat guano, the DNA in the bat guano, and compare that using metabarcoding and quantitative PCR, which is species specific, but much more sensitive for 3 different insect pests. So that's another chapter that Erin's working on. But these, we're basically taking the bat data set and then doing another analysis to look at another question.
Very good. Yeah, and I neglected to mention that, but this was kind of a neat project because, like you said, Brooke, you had a CIG previously that was at a different scale, and then we went to this one. So it's kind of neat how you've built off some of your past success to this one, which I thought it was fascinating when I was reading your final report. So, Edwin, you've got your hand up.
Thank you, Edwin. I have a question on did you include like a farm pest management as part of one of the variables? And the question is if there was a pest management system in place for the farm, and the amount of food for the bats and the barn swallows are not there, how far they can travel to find some food source.
That's a good question. Some of the farms I worked on had IPM in place. I think 3 of them did already for brown marmoset and stink bug. And we still had plenty in our traps. And I guess you're talking about if there was an IPM strategy where they can mitigate all of the insects that are pest species. To be honest, the current pest management that we have now would never get to a point, I don't think, where it would wipe out all the insects in that area, because even some of the farms I worked on, I still found a ton of brown marmoset stink bugs in my trap. They had them in their traps. And a lot of times the farmers were spraying for insects as well, and we still had them. So in a hypothetical situation where the pest insects were no more, the bats would still have plenty of food because there's so many insects that they could just take from. As far as how far they can travel, bats can usually travel around 5 kilometers or so in a radius from their roosting site where they maternity colonies, for example, where they have their offspring. They can travel quite a bit into other areas, but I don't think any type of IPM strategy would mitigate their food source realistically. Does that answer your question?
Yes, it did. Thank you very much.
Of course, thank you.
All right. Last call for Erin or Brooke. All right. Well, thank you both very much. I thought that was really interesting. So, yeah, great job with that. And at this point, too, we'll just open the floor back up in case anyone thought of any other question. Oh, and I should say, Brooke and Erin, if you're okay with it, do you mind posting your contact information in the chat in case anyone thinks of something they want to follow up?
Sure.
Cool. Thank you for that. So any other questions going back to whether it's a Manure Link or John Spartina project or anything else like that? All right. Well, seeing no additional questions, I really want to thank all 3 of you, 4 of you technically, for being on here today. Like I said, I think it was just fortuitous that kind of all 3 of these wrapped up around the same time, and I was like, man, this is a neat spread again of kind of what CIG can fund and what we can do. And I thought all 3 of your results were pretty neat, and definitely wanted to get that shared out to our staff and our conservation partners. So kudos to a job well done on all 3 of those CIGs for sure. And I appreciate you guys taking the time to kind of share your findings from that. It's a big part of CIG. We want to get the word out, make sure people are aware of it. So thank you to all 4 of you for being here today. And like I said, I think it was excellent. So good.
Thanks, Evan. We appreciate the support. I mean, it's a nice network that we have here in New Jersey. So it's good to be able to share all of our results too and work with everyone. It's a lot of people, but it's a lot of good people,
Yeah, yeah. I guess it's good being a small state too. We'll get to collaborate a lot, So
Yeah.
it's good.
Yep.
Good. All right. Well, I guess with that, we're actually gonna get a half hour a day back for this too. So it's good. So yeah. So thanks again. I'm sure Brooke will be in touch. John definitely. Sandy, I talk to you a lot too. So we'll all be in touch. But excellent. Yeah. And thanks for our colleagues from some of their states here for showing up too. So we're happy to spread the word there. I think there was a lot of overlap when you look at Delaware and Maryland and some of these other states. So it was good. Awesome. So thank you all so much. You guys all have a wonderful day. We did record this and we will get it posted and out there and available.